So hello everyone and welcome, and thank you for joining us for what I hope will be very interesting and informative discussion today around some of the latest data in rheumatology. My name's Professor Peter Nash from Griffith University in Brisbane, Queensland in Australia, and today we're very lucky to be joined by Dr Ameen Jubber, Specialist Registrar in Rheumatology at the University Hospital of Leicester in the UK. So hi Ameen and welcome. Thanks for joining us today. And we're going to be discussing a really very important issue today, a recent editorial paper of Ameen's reviewing the evidence for venous thromboembolism risk with the JAK inhibitors and whether this risk also extends to the more JAK1 preferred or JAK1 selective JAK inhibitors. So this is a paper that's been published in Expert Opinion on Drug Safety. It recently published online, 13th of July. So let's start, Ameen, by just telling us a little bit about yourself. Where have you come from, where are you working, and what are you working on?
Well, thanks for the invitation. So I'm a registrar in Leicester, which is in the East Midlands of the UK. I work in a big tertiary centre, and in our department we prescribe a lot of JAK inhibitors. And so in the UK we have 4 JAK inhibitors that are approved. There's tofacitinib, baricitinib, upadacitinib, and filgotinib. And they've become more and more popular in recent years, especially to patients who are not keen on having injection therapy. So they've become a very popular option. And it's important that recently NICE, the National Institute of Clinical Excellence, has approved using upadacitinib and filgotinib in patients with moderately active rheumatoid arthritis. So these are now accessible at a much earlier stage of the disease for many patients. But we also take into account the results of the oral surveillance study, which led to a black box warning for tofacitinib, but they're also extended to baricitinib and upadacitinib. And so because of some overlap in the mechanism of action, and so the EMA now advises giving tofacitinib to patients over 65 only if there's no viable alternative. So we wanted to assess the available data for VTEs in the JAK inhibitors to see if it's a class-wide effect, to see how that will impact on our prescribing.
Okay, so since oral surveillance, 2 questions. Has it affected JAK prescribing in the UK? And I'm sure the EMA review that's about to get published soon will certainly affect JAK prescribing. So have you noticed a change in prescription? And is there a tendency to, what's the market share like in the UK? Is it more UPA Filgo, or is it still pretty much across the board, all 4?
I don't know if, the numbers for that, but in terms of whether it would, how it's impacted on the prescribing, we go by the EMA's recommendation there. So what's come out from the data and looking at the other JAK inhibitors is you have to take into account the patient's cardiovascular and VTE risk profile. So the patients that were included in oral surveillance were over 50 with RA and had at least 1 cardiovascular risk factor. And it was the 10 milligram twice a day tofacitinib regime that led to that significant imbalance in PEs and all-cause mortality, and that led to that treatment arm being discontinued, and that led to the FDA and EMA recommendations. But for example, if you look at the US Corona RA registry, there they compared tofacitinib 5 milligram twice a day with other biologics, and they didn't find any significant difference in VTE incidence risk. For example, in the tofacitinib group, it was, I think, 0.29 per 100 patient years, and in the other biologics, it was 0.33 per 100 patient years. And in another post hoc safety analysis, they found that among patients with receiving tofacitinib at all doses, in RA, psoriatic arthritis, and psoriasis, those who had cardiovascular risk factors had a much higher risk of VTE than those that didn't. So we have to take into account the patient's risk profile. But as you mentioned as well, because with NICE we take into account their recommendations because it has impact on health economics. And so because NICE now approved upadacitinib and filgotinib with a lower disease activity score, we think that in future we'll see more prescriptions of those agents.
Yeah, interesting, and I think there's more data to come. We were always concerned that the FDA has made a blanket class decision for every disease indication based on one head-to-head with one drug in one disease indication, and the other patients with other diseases have much lower risk. But anyway, look, let's talk about the British registry, which is quite large, been going for a long time. They did give the background rate of VTE with TNFs, and from memory, it was 0.6 to 0.8 per 100 patient years. That is kind of a background TNF rate, if you like, very different to what they found in surveillance, but we can't ignore the fact that there was non-inferiority with a good margin. So let's talk about VTE. Now, give me a feel, before we get to your paper, what you think mechanistically the JAKs why could they cause VTE? Because I've never seen published, and it might be because I missed it, but I've never seen any of the basic coagulation profile data published with JAKs. Von Willebrand factor, factor V Leiden, protein C, protein S, antithrombin 3. I've never seen any study where they looked at the effect of JAKs on those elements that we would normally look at in a procoagulant situation. I've seen something with platelet counts, but I've never seen any of that basic science. Are you aware of any studies that might give you a mechanistic reason, other than disease activity and high-risk patient population, that these patients would be prone to VTE? Lupus anticoagulant, anticardiolipin 3, you can think of lots of things that should be tested. And we've certainly put protocols to the companies, but there's much resistance in doing them. Are you aware of any data with those things?
No, I'm not. But it's very interesting. I mean, it's one of those things where more work needs to be done to figure out the mechanism of how this happens, because some of the other side effects of JAK inhibitors, like especially viral infections, it's better understood. But with the VTE, there's a lot of work needs to be done to figure out, is it an on-target or an off-target effect? And it seems counterintuitive that patients with RA who have a higher background risk of VTE, given the fact it's an inflammatory condition, it seems counterintuitive that when you reduce that inflammation, you actually see a higher VTE risk. So no, more work needs to be done in that regard.
Okay, so let's have a look at your paper then. Can you tell us a little bit about how you went about doing it?
So we looked at all the major studies looking at background risk, looking at the risk of VT. Of course, they also include the safety profile as a whole, but we zeroed in on VTE risk to make it to just focus on that section, because in terms of VTE, we know this is a potentially life-threatening thing, and it's important to take into account, as much as it's important to take into account efficacy, you have to take into account safety. And patients nowadays, they will approach and ask you these questions. So we wanted to have a good understanding of what the data shows, what are the pitfalls of the data. So it involved a literature review, and we focused on the main JAK inhibitors used in the UK. We also briefly mentioned peficitinib, even though that, from what I understand, is only used at the moment in some of the East Asian countries. We don't have it. We don't have it in the UK, yeah, but we mentioned that one briefly.
Japan mainly.
Yeah, but what we found in most of the data available was for tofacitinib and baricitinib, and there are much less patient years of exposure available for the for upadacitinib and filgotinib, and that's one of the things we highlighted in our results, is that it's perhaps, although the data looks more promising for those agents, it's too early to draw conclusions that they're risk-free for VTE.
So let's just have a look at some numbers here. MACE is a separate issue. If we look at the VTE rates with TOFA in surveillance, and we stick to the 5 milligram, what was the risk of TOFA in the rheumatoid population over 50 with a risk factor? It was something like double or triple compared to a TNF, but the actual rates were pretty low for both, and very low for the TNF.
Yeah, exactly. Yeah, and like we said before, those patients who were there, over 50 with at least one cardiovascular risk factor. So that needs to be taken into account as well.
All right. So the actual rates I've got the numbers here. 5 milligrams VD compared to a TNF. 0.3 was the incidence rate with TOFA, which seems less than the TNF in the British registry by half, and 0.18 for the TNF. So they had 10 cases in nearly 1,500 on TOFA, and they had 6 cases in nearly 1,500 on the TNF. So, these things are not common, and there's a difference when you look at large populations, but it was a head-to-head study, and therefore I suppose we can't ignore it. There was differences in baseline characteristics like smoking and all kinds of other things, but that's a separate issue with lung cancer and MACE. But okay, so tell us a little bit about your findings, and in particular, whether there was a difference that selectivity you think will make or not make, or you just don't know yet.
So with the second generation agents, so with upadacitinib, most of the data comes from the SELECT phase 3 clinical program, and they looked at 15 milligrams and 30 milligram regimes, and they compared that against placebo, methotrexate, and adalimumab, and they found similar VTE rates across those groups. But interestingly, when they looked at the VTEs that occurred in the upadacitinib patients of all doses, they identified the risk factors as prior VTE and raised BMI. Then for filgotinib, we looked at an integrated safety analysis, and they looked at both the 100 milligram and 200 milligram dose regimes, and they found incidence rate in the 200 milligram regime of 0.2 and 0 in the 100 milligram regime.
And you think that means that the filgo might have less than the others, or just that over time there's selection bias and you don't put people into these phase 3 trials who are at high risk of VTE?
Yeah, exactly. They often exclude patients who have high risk of high risk cardiovascular disease, but also sometimes what happens is if you only look at the placebo-controlled part of the trial, potentially you can miss the VTEs that would happen in the long-term extension part, and so that can lead to an underrepresentation or an underestimation of VTEs.
The other thing I recall, the TNFIR study of upadacitinib in the first 24 weeks showed that they had 0 in the control, and they had 4 events out of 200 people followed for 24 weeks. And they were most concerned because there's PEs and all kinds of things. But then as they continued that study, those risks disappeared. And when they did their head-to-head against the TNF and methotrexate, the VTEs were nicely shared amongst the 3 groups. So do you see any hint from your data that VTEs increase over time with any long-term follow-up? Because if you think mechanistically, then there should be more time, more exposure, more events.
Yeah, and We haven't seen data that definitely proves that it happens later on. But if for example you take if I give another example with baricitinib, when they did the placebo-controlled trial in the first 20, I think it was the first 24 weeks, the baricitinib group, out of 1,000 patients, there were 6 VTEs, and that was just in the first 24 weeks, and there was 0 in the placebo-controlled group. But then when they looked at the data, they found that all 6 of those patients had VT risk factors. When they were switched over, and those patients in the placebo group had baricitinib, there were 0 VT. So those results weren't replicated. So yeah, that makes sense. It could well be that it's something that happens in the early stages, but then you can't, because we expect many of these patients are going to be on these drugs for a long time, or we hope they will. So we need to take into account the long-term effects.
Well, we have seen with infection and probably with VTE risk that those at risk have events early and drop out, and the people without risk continue happily on their medication with the control of the disease. So that's a bit difficult. In one of the studies, meta-analyses, COX-2 inhibition was considered a risk factor. Is that your finding from this study?
In patients on upadacitinib, when they looked at the when they looked at the 15 milligram regime, I think they found that NSAIDs were a risk factor, but I don't recall COX-2, but I have seen that meta-analysis, yeah, where they mentioned that, yeah.
Okay, so what take-home message would you like rheumatologists to take from this editorial? In particular, how should we manage patients with and without a history of VTE? How do you manage them?
Yeah. So I think we still need to be open-minded. Obviously, more data will come with time, and in the meantime, we say that clinicians have to make an assessment of the patient's VT and cardiovascular risk profile for all the JAK inhibitors, because there's still we still need a greater understanding of the mechanism of how the JAK inhibitor could potentially lead to VTE. So we don't say that we think it's too early to conclude by looking at the current data that the second generation agents are free from risk. It might be that as more data comes with time, we may see that it is in fact a class-wide effect, or it may not be. But given that VTEs are very potentially life-threatening, we have to be open-minded.
And say you have a patient, which we all have, who failed everything, and JAKs are almost their last port of call, but they've got a terrible history of recurrent VTE over the years. How do you manage that person?
So it would be a risk-benefit discussion with the patient, but also if the patient is on an anticoagulant, you know that some of that risk is mitigated. But then there are other sides to it as well, like potential cardiovascular events, which may not have that protection from being on an anticoagulant.
So have you ever had to anticoagulate someone who's got no other choice but to go into a JAK?
That I've not seen before, no.
Right. Well, I certainly have. Okay, so take-home message: weigh up risk and benefit. More data needed to understand from a mechanistic side, because I'm not sure that's been done very well. And we need real-world data like Corona and the Swiss and the Swedish and so on. And are patients on JAKs being enrolled into the British Society of Rheumatology Biologics Register, or is that closed?
I'd need to check that.
Yeah, because we need that kind of data to help us, even though there'll be all kinds of issues with selection bias and who goes on to what drug and time in their journey as to what drug they've left with and what they've already tried, etc. But those kinds of things will be very important. Anything else you want to mention about this particular issue going forward? Any future research you're going to do?
No, but like you said, a very good marker is retention, and that's something that we could look at in our own cohort of patients, is the retention on JAK inhibitors, because it gives you a very good marker of adverse events, but also how well the patient is tolerating it and how well it's working. So we have a very good database in our hospital on patients on each biologic, and that's something that we could look at.
So thanks very much. Thanks again for your time, Ameen. If you'd like to know more about this paper and others uploaded to the CSF website this month, you can get detailed slide sets are available in the publication section at cytokinesignalingwith2ls.com. Please subscribe to this podcast on iTunes or wherever you get your podcast media. Give it a review and let us know what you think. Thank you very much for your attention.
Thank you.